Hi, Keith here with another of my statistical videos, this time in the series on PAST, the paleontological statistical program that's becoming more widely used in other disciplines. And up here you can see how PAST looks, or actually how it looks now, because this is version 3 and the look and feel and operation of this is somewhat different from the previous version, although all the same statistical methods are still implemented. So for this demonstration we need some real data from a study we did some time ago, looking at how the intensity of light affects the growth of mangrove seedlings. And so this was done in cleared areas created by cyclones, and let's see little seedlings in through here, in the fringe there, and then deep in the forest beneath these trees where the light intensity can be quite low. And we had areas shaded by shade cloth which cut out 80%, 30% of the light, and then unshaded areas. And that work is published here in this manuscript. I will say that the species is no longer called Cereops taggle, it's called Cereops australis. There was a revision of the taxonomy of this, and we refer to the seedlings as propagules because they're different from ordinary seeds. Okay, in the old days we would select, the old days of PAST, we would select the data and copy and paste. Now before I do that, air temperature, soil temperature, relative humidity, light, the moisture of the soil or moisture content of the soil, and salinity parts per thousand, which can get high because the salty water moves into the mangroves and gets evaporated and leaves behind the salt. And over the side here we've got CL for clearing, FF for forest fringe, and FO for forest, and obviously 0, 30, 80 for the different amounts of light, the different shade cloth cover. So, Control C to copy over to PAST. What I would do is edit mode, edit labels up here, and edit paste, and then turn those off. Now what I'm doing here is I'm assuming you've got labels for the columns and labels for the rows, which is my recommended procedure. And then everything's fine in here. We can drag these around if we need to move them around, and we can drag and select to pick up the data we want to work with. But I'm not going to work with PAST anymore. PAST 3, I've got select mode on, and I need to check these boxes so I can see and modify the attributes for the columns and the rows. I'll click in here again, edit, paste, and we're looking pretty much the same as for PAST 2. If I want to move columns or rows around, I need to click here. What I can see here different though is row and column attributes. Now for the columns, we can now set a particular type of data. I'm not going to bother with that today. And down here for the rows we can set different symbols and different colors. If you try to do this, you'll find it's still a little bit buggy, and there are parts of PAST where there are some bugs. So I'll alter some of these, and you see nothing happened. So try it again. I will do this just for the purpose of the grass red. And I've got the colours changed but not the symbol, so I'll do here for the other group, and let's change to blue, and I'll change this, and let's change some things but not others. Now let's get rid of those. Now with these environmental variables, what I would like to do is a PCA, so I can look at how the environment varies depending on the different experimental treatments and locations. So I'm still in select mode, and I've select and drag there. So that's still working. Now what I need to do first, usually, is transform. Because, as I've talked about in other videos in this series, these environmental variables here are different kinds of things. Air temperature and soil temperature are the same kind of thing. They're measured in degrees Celsius, but relative humidity isn't, light isn't, moisture and salinity, they're all different kinds of things. And if I rescaled air temperature to be degrees Fahrenheit, I'd get different results, which doesn't make a whole lot of sense. So I'm going to go in here and evaluate expression, brackets, current cell minus mean, close brackets down here. Now you see the cursor, this is one of those little buggy things, the cursor's showing up there but the bracket is going on at the end, and divided by standard deviation. In practice I might copy these data into another set of columns and transform there so that I could also keep the original values to look at, but I'm not going to bother here today. Done. Close. And now select again, multivariate, and you'll see the big long menu from PAST has been reduced here into a set of drop-out menus, which is a lot nicer and a lot easier to work with. So ordination, principal components. Now I'm running a little program which allows me, it's called Sizer, which allows me to resize windows to particular sizes. So I'll make this big. So here we've got the results of PCA. I'm not going to fiddle with any of this stuff over here. We can go to the scatter plot. And what's different between here and PAST version 2 is that now the results come up as tabs instead of different windows. And again, that's a little easier to work with. So if we go to the scatter plot, here's our PCA scatter plot. We can put on the row labels, we can put on the biplot to show the vectors, and graph settings is now here a button instead of right click, so graph settings. Now you'll notice up here some of this text is not displaying properly. That's something weird to do with my computer and I don't know how to fix it. So I change the size of the symbols, put on some thick lines, and there's the PCA. And we can see here that the clearing, the forest fringe, the black symbols and the blue symbols quite clearly separate out, and there's an axis going this way which is to do with increasing moisture, which is the damper soil as we get in under the forest canopy because the light doesn't get there, and so the seawater evaporates more slowly. Going across the other axis, left to right or right to left, we've got relative humidity, light levels, soil temperature and air temperature. So the shade cloth there is affecting the micro environment in those different treatments. And this is not exactly perfectly how we want it to go, but it is pretty good because we were trying to separate out different environmental effects, and the treatments here worked. Going through the other tabs, the scores here, these are the scores for each of the results. These are averages, I should say. The original data are averages for all the replicates and the different times we sampled and over the two clearings. Here's the loadings plot. This is looking at PC1, so we can see that this is a contrast between air temperature, soil temperature, light levels and salinity, and on the other hand, relative humidity and moisture. So it's really two different sorts of related things going on there. And if we select PC2, it's mostly moisture, salinity and air temperature, with the other variables not playing much of a role. The loadings, so these are the numerical values that give us the plot here, and lastly the scree plot. So the first and second of the two axes take in a fair amount of the variation in the data. There's also some in the third axis, but and fourth axis, but after that there's not too much. But we can be comfortable the first two are capturing a reasonable amount of what's going on. We've got copy here before as previously, so I can copy this and then paste it into a document or print. And that's where I'll stop with this. All I was trying to do here was just introduce a new version and show you how to get the data in and do a little bit with it. The previous videos I've done will still be valid because the basic methods here haven't largely changed, and if I click I can do what I did before. Okay, thank you, and I hope that's helpful.